Welcome Marcin Krzykawski on the stage.
Hello. I need to get used to standing here and seeing all the lights. I'd like to tell you a story about a company. It's a story of failure, but later a story with some glory at the end. Let me tell you how it all began. Some time ago, about like 5 years ago, I visited a friend in Portugal, in Lisbon. And I was doing my PhD in biotech, he was doing his PhD in biotech. And that evening, I told him that I think I got to the point where I can grow a tumor, a cancer tumor, that is 1 centimeter in diameter. And for those of you that don't know, growing such a large tumor is really hard, it's really challenging. Obviously, my friend told me he doesn't believe in that. I thought I believed that, so we bet on it. And we actually, what we bet on was a nice bottle of port wine. And I was quite well motivated. So I went back to Poland. For half a year, I worked on this project as a side project. And yeah. The problem is that after those 6 months, I realized that I failed. And I had to give the bottle to my friend, and we just moved on. But that's not the end of the story. That's actually the beginning of the story. Because the most important thing is not that I failed, but the most important thing is that actually I learned why I failed. And I also learned that most of the big biotech companies and pharma companies are facing exactly the same problem or something very similar to what we face and why we actually failed. And we decided to challenge the problem and to work on the solution. So first, let me tell you some things about the market, about what is actually going on today with drug research. Did any one of you ever thought how new drugs are being created? Yes, you did. Okay, you should be knowing that. But the majority thinks that new drugs are made in the way that somebody in university is actually finding a perfect drug for something, then they patent it. Then they sell the patent to the company develops this patent, and then you can buy drugs in your pharmacy. But the problem is that it is actually totally different. In real life, companies start with they actually start by deciding they are going to look for a drug against certain disease. So they start with about 10,000 different drugs. Through the research process and different types of simulations, they actually narrow it down to about 1 or maybe sometimes 5 possible drugs that may be working. And then with those 1 to 5 drugs, they actually go to clinical trials. And it would be okay if there wouldn't be some few, actually 3 major problems. The first problem is that this whole process from the very beginning to the drug in a pharmacy takes about 10, sometimes 15 years, or sometimes even more. This whole process costs about $1 billion, and actually recently I found one drug that cost $2.6 billion. It takes so long, it costs so much, and at the end you have the probability of finding the right drug less than 5%. So you can spend a lot of money, make a lot of research, and still you have between 1 and 5% of really finding the good working solution. And that was our motivation, and that's what made us think that we should do something about it. So let me introduce you to our first product, LifeGel. LifeGel is a hydrogel for 3D cell cultures. But what are actually cell cultures about? Cell cultures are this kind of tool that you use when you narrow it down from 10,000 different drugs to 1 or 5 different drugs. So you need to investigate the drug you're working on and to find which one of them is working the best. So, and you can do it in 2 ways, 2-dimension and 3-dimension, that I will talk about later. But sorry. But I'd like to, but LifeGel is actually the improvement that you can make in the process of research when you narrow down the number of possible drug, let's say. So what you can do with it, actually with LifeGel, you can improve the effectiveness of your drug discovery, so the probability of finding the right drug will be higher. You can also support the efforts for better science, so if you're not looking for new drugs and you're doing some other kinds of research, you will also get much more valuable results. And we hope it will allow the transition from the global two-dimensional cell culture market to 3D, but I will talk about it later. So why is it important to change the two-dimensional way of researching new drugs to three-dimensional way? Imagine somebody living in a society, surrounded by friends, family. You can communicate to those people, you can interact with them. And then imagine taking this person and putting him on the desert, without friends, without family. And this seems broken. But the problem is that what we are doing in science is actually very similar. You take one cell from somebody's body, you transfer it to a flat plastic dish where it cannot communicate or behave as it would behave in real life, and then you make research and make serious decisions based on how this cell actually responds to what you are doing. And this process has actually led us to development of hydrogels, of like external scaffold for cells that allows them to grow as inside the human body. And this way we work to recreate the real environment that you can find in your body and to make the research on you know, this kind of experimental setting. And so how it looks actually. Well, it's not going to be so well visible, but if you would look at this slide, you could actually see a bunch of flat two-dimensional cells because they just live on a flat plastic dish. But if you transition to 3D cell cultures, I'm afraid that you cannot see this so well, but you can clearly see that this is actually exactly the same type of tumor, but in this way, you can actually see the tumors growing. And if you investigate something on this kind of setting, the results you get are much closer to the real life. Like with another example, if you, no, this is not going to be visible, sorry. So, why is it so important? Why people are not doing this all over if it's so obvious? This whole cell culture process started actually somewhere in 1950s. And ever since, it has barely changed. Like in 1950s, 60s, or 70s, it was actually a very useful tool because we didn't know anything about it. But since about 2000, we already know, and there are more and more evidence showing that you should transition to 2D, to 2 dimensions, but because of various reasons, many people actually don't. But there is a trend, and I want to show you the trend for the revolution that is coming actually in this field. Because back in, like, okay, it's not here, but because like 10, 15 years ago, less than 0.1% of the whole cancer, or not only cancer research, but cell research done on cell cultures was done in 3D. Now it's about 2%. So it's not much yet, but it starts growing rapidly. And you can see this rapid growth more if you look at the market size, where in 2013, the market size was about $500 million, and in 2019, it's expected to grow to $2.2 billion. And for us, this shows the clear trend and clear revolution of what is actually going to come and the benefits we hope to get from that. So, are we the first ones? So, who is there on the market? Well, we are not the first ones because somebody has done a lot of research with that data that I just showed. But we have, with our product, what we actually concentrated on to improve on the mistakes the other products actually are making. And the first improvement for us was actually the clearness, the clarity of the product. Because if you do research, actually most of the, actually all of the competitive hydrogels for 3D cell cultures are contaminated with proteins called growth factors. And if you are doing research, growth factors are actually influencing very heavily the results you're getting, because they stimulate cells and they can actually alter the whole experiment just because they are contaminated. The other very important skill argument supporting LifeGel is that LifeGel is extremely repeatable. It means that if you make the same experiment over and over again, regardless whether you do it in Kraków, New York, or Shanghai, it's going to be the same. Whereas when you buy any of the competitive hydrogels, you're actually going to get different results because the different production batches are going to be different. Like when they produce it in March, it's different when they produce it in November. And last but not least, and it's actually more important for the business side, is the scalability. If you want to scale the competitive products, you need to have investments just to scale your production capacities by like 20 to 30%. For us, it's quite easy to scale the production to supply big part of the global, supply. So what are we going to do? Until now, we have developed hydrogels for 3D cell cultures of tumors that allow us to investigate how they work, and the pictures I showed you were actually our own data. And we also developed a test for angiogenesis that's extremely important in cancer research when you are looking for a treatment that will actually cause those blood vessels to shrink and kill the tumor in that way. But we are working on new applications that I cannot really talk about at the moment. But we are also planning to go to stem cell research, neuronal cell research, artificial meat research, artificial tissues, individual drug testing for individual patients, and many others. But this is just a kind of vertical growth. We also plan the kind of perpendicular growth where we will create new products that will actually allow to automatize and visualize the research you can do with LifeGel. So who we are? We are a team of excited people, but until now there are just two of us, me and my co-founder Renata. I have about 12 years of experience in laboratory work and business trainings like Cambridge University Ignite training or Kaufman Foundation Fast Track. Boom, boom. Okay. And Renata has about, well, something is changing. Hello, hello. Renata has a master in biotech, a lot of experience in laboratory work, and even more experience in, working in the company and developing the company itself. And we are a team of two, but we try to work together so that we will complement each other's skills. And with the skills that we don't have, we have a bunch of outside advisors, both from scientific as from the business side. And yeah, we are working together towards the same goal. So that's more or less this. Our company's name is Real Research. We wanted to name it after something that's really our goal, to make the research real. And our first product is LifeGel, and with LifeGel we want to change how science is being done. If you want to contact me, these are my data, my contacts, or talk to me after the presentation. And one more thing. Today I think I would have won this battle. Thank you.